All right. Hello, everyone, and welcome back to my channel. My name is Dr. Anwar Yusuf Dunbar, and this is Big Discussions 76. Let me turn that down. This is Big Discussion 76, Science and Technology. Today is January 23rd. I haven't done anything over here in a while. Just it's been a busy new year getting started. So I'm back with another live broadcast discussion regarding glucose 6-phosphate dehydrogenase deficiency. What's interesting about this particular stream is that I'm also streaming over to Facebook. So this is my first time streaming from this channel over on Facebook. I don't share all of my content on Facebook, but I thought this is an important topic as it's a human health issue, and it's relevant for what's happening in the world right now. And so what I'm going to do is I have my guests backstage, but before I bring them up, I'm going to give a short walkthrough of how I became knowledgeable in glucose 6-phosphate dehydrogenase deficiency. So, so how did this all start for me? Well, it started for me like it started for everyone else at the beginning of 2020, or should I say in the first quarter of 2020, the COVID-19 pandemic started. And that was right around the time when I started this channel. So I thought that with this being current news and a current event in a rapidly unfolding pandemic, I thought that it would make for very compelling content to cover here on my channel. My background is actually in pharmacology, so the study of drugs and what drugs do and how humans, the effects of drugs on humans. And so one of my professors at the University of Michigan once told us that pharmacy is the study of what drugs do to man. Pharmacology is the study of what man does to drugs. So my interest in this pandemic initially was from naturally the pharmacology standpoint. That led me to start asking questions about what were the potential effective medications for COVID-19. And that led me to two controversial drugs, chloroquine here, which is this structure. This is an older drug, which was used for malaria in Africa, and I believe in the Mediterranean regions. So my curiosity led me to this, and also the science community was interested in this initially for the treatment of COVID-19. And then I was led to, my curiosity led me to this, and this was also in the news as a potential treatment for COVID-19. And I won't get into the particulars about it, but there was evidence. I did do a video on this channel citing some evidence that it was effective in decreasing the virus's multiplicity. So I'm going to leave that there. But then it was brought to my attention that there was a population group who had potential adverse reactions to these two drugs, chloroquine and hydroxychloroquine. And that population group turned out to be individuals deficient in the enzyme glucose 6-phosphate dehydrogenase. Just give me 1 second while I bring the picture up. There it is. So this is the enzyme the enzymatic pathway for glucose 6-phosphate dehydrogenase. I think Niloufer will like this one a little bit more. I think the one I was using before, might have been in a foreign language, but this one says glucose 6-phosphate dehydrogenase. So I think she's back there laughing. But basically, what happens is the glucose 6-phosphate dehydrogenase catalyzes the production of NADPH from NADP. I believe this is the pentose pathway, and that NADPH gives rise to glutathione disulfide. And so glutathione disulfide is an antioxidant. It's a natural antioxidant in our cells, and our cells have antioxidant systems which protect our cells against oxidative damage. In this diagram, you can see there at the bottom hydrogen peroxide, and hydrogen peroxide is a byproduct of enzymatic reactions of all kinds. And if this hydrogen peroxide builds up too much in our cells, we can deplete our natural supply of glutathione, resulting in oxidative damage in a given cell type. So it turns out that there is a subpopulation who are deficient in this glucose 6-phosphate dehydrogenase. And these individuals, I thought I loaded all these pictures up. Let's see. There it is. Looks like Nicole Ali's here. Good morning, Nicole. I know it's early out there on the West Coast. Thank you for stopping through for however long you'll be here. So those individuals who are deficient in glucose 6-phosphate dehydrogenase, they are susceptible to something called hemolytic anemia once they fall under oxidative stress due to drugs such as hydroxychloroquine. This is dangerous because, as you know, our red blood cells, those carry oxygen from our respiratory tract to the other organs in our body. So the red blood cells carry oxygen to our other organs, and they carry carbon dioxide back to our respiratory tract to expel, so we can exhale that carbon dioxide. So if you have destruction of those red blood cells due to oxidative damage, well, the person is going to get very, very sick. The last thing I'll say about this is that this is a pharmacogenetics story for context. And so what pharmacogenetics is it is a subdiscipline of pharmacology. And actually, it's a subdiscipline of toxicology. And basically what this discipline is that it's looking at the genetic, the individual genetics of the patient. And so it's getting us towards individualized medicine. And so the theory is that we're not all genetically the same, and that we express certain enzymes differently. And this is important when you're thinking about drugs, because there are enzymes that help the body handle drugs in a certain way. And depending upon the levels of your drug metabolizing enzymes or your antioxidant pathway related enzymes, differences in expression of those enzymes can cause differences in how your body handles those drugs. I hope I communicated that effectively. So that's why we're here. We know that I'm going to bring up one more picture to illustrate this, then I'll bring my guests up. Yeah, I like this picture here. This is a picture of the nucleotide base pairs for DNA and RNA, but this is DNA because I see the T there instead of the U, the thymine instead of the uracil. So it's about our genetics. We're not all the same. And because of our genetics, we handle drugs and medications differently. And there's a subpopulation, a significant subpopulation, which my guests are going to tell us about, who are deficient in this enzyme, glucose 6-phosphate dehydrogenase. And for as prevalent as this deficiency is, it's not well known. So I'm going to bring up two of my guests, Keely and Nalufer. They've been on before from the G6PD Deficiency Foundation and Taskforce. I'm going to bring them up first. Good morning, Nalufer.
Good morning, Anwar. Great to be back.
Yes, good morning, Keely.
Good morning, Anwar. How are you today?
Good, how are you?
Good.
Okay, so do you ladies want to say any words before we get to the PowerPoint presentation?
Sure.
And remember the rules when you're not talking.
Mute.
I'm going to mute right now.
Yeah. Okay, so since Keely muted, I'm going to start. So thanks so much, Anwar. You're right. I did like that picture a little more. I am, I don't know if you want to call me the founder, but I'm the initial member and part of the task force that we started on G6PD and COVID to try and get more awareness about the deficiency and the risks to this population during the COVID pandemic. I myself am G6PD deficient. I have a lot of family members because it is an X chromosome linked deficiency. So I have a lot of family members that are deficient as well. And I've been working very closely with a lot of amazing people around the world, including yourself, Anwar, and Keely from the G6PD Deficient Organization to spread this awareness. So Anwar, I must say we've had 3 sessions with you so far. One was around just introducing the task force and the conversation around G6PD. The second one was on safe and unsafe drugs. And the third one was on the etiology. And of course, this one is very interesting as well. It's about testing and testing around G6PD and the relevance of it during the pandemic. So I'm very excited. We have excellent speakers, guest speakers on today, very impressive resumes and biodatas, I must say. And I'm looking forward to the session. And all I can say is that, so far, Anwar, 2021 is looking very, very positive. I think we have started to get the word out. We're doing a lot at the state level in the US, and there are a lot of research papers that have now come out, including yesterday, I saw something that's come out on the WHO website, which is what we've been very keen on working with WHO. But I did see, of course, parallel to what we're doing, but they have loaded in some research papers around G6PD and the importance of it during COVID. So I'm extremely positive in 2021, hoping that we can really make a difference. So thanks so much for having us, and I won't take up too much time. I know we have a lot of content today to go through.
Okay, yeah, I'm sorry in my intro, I forgot to say, yes, we talked about the drugs and we talked about the pathway and it just, but you presented it nicely. So, okay, so Keely, I think you're up next. So I have your presentation here. Do you want to say anything before we start the presentation?
I just want to thank you so much for this opportunity. It's wonderful. Thank you.
Well, let me cue up the presentation and I'll hand the microphone over to you. So.
Okay.
Okay, give me 1 second here, Keely. This thing is finicky. Let me close it and reopen it.
Do we want to introduce the other 2 guess? We could do that.
Do you all want to do that now? I mean, I don't want to. In terms of connectivity and.
Okay.
Yeah, let's get you started first.
Okay, so this very first screen is talking about the concerning prevalence of G6PD deficiency. And as you know, glucose 6-phosphate dehydrogenase deficiency is one of the most inherited genetic enzyme disorder in the world. It's usually referred, at least I will refer to, as G6PD and then a small d. The majority of the times, those who have the deficiency have no obvious clinical symptoms and are often unaware that they even have it. Looking at this map, you can see how it is really widespread worldwide. I'm particularly interested in USA. That amount is between 4 and 7%. But I am also concerned about the whole world. It is amazing how it is so widely spread. And in fact, I know the WHO had a map, and you'll see in a lot of the older papers, when I mean older, only a couple of years older, in 2000, no, when was it? In 1989, they did one, and the amounts were much less reported than what is on this map, which was about 2010 reported. So it's not that it is growing. I think it's because it's being more screened properly, or it's being more screened or thought about. I really don't understand the discrepancy of the difference, but it could be, that could be an indication. Let's see, the WHO, which is the World Health Organization, recommends G6PD universal screening if 3% of the male population is deficient. It is the most effective for G6PD deficiency screening for neonates born in multi-ethnic, multicultural society settings. And this was stated in the American Society of Hematology in 2016. So what they're explaining right there is pretty much United States and probably other areas as well. So it is recommended out there in these papers or journals and so on. But obviously, we are behind in that in the United States. We do not really screen for this deficiency, and a lot of people have never even heard of it.
And Keely, I'll just add that I think, like you said, Keely, we're behind. I'm sorry, there's a lot of disturbance. But we're behind in the world, across the world. And I know Tivani is with us today, and she'll talk about it from a South Africa perspective and just the African continent perspective. But it's surprising. I just had someone reach out and is a new member of our task force. And he was mentioning he's from Greece, which is where it is very popular. And nobody that he knows has any awareness of it. And the fact is, these numbers are so high in all over the world is significant. There are 500 million. The research papers say that 400 to 500 million people are deficient in the world, and yet hardly anyone is aware. And unless we get this more testing done and more awareness created, it is a huge challenge worldwide. Sorry, Keely didn't want a button, but.
Can we go to the next slide, please? This was already brought up today from Anwar, and it was wonderfully explained. I just quickly did this because a lot of these things are going to come up later in regards to screening. So I think this is important that everybody really understands the biochemical pathways involving the G6PD enzyme. So as it was already pointed out, the pentose phosphate pathway, better known as PPP, sometimes I've heard it as a shunt. You know, there's different terms for this, but the PPP, the most interesting part about this is that it is accounted for approximately 60% of that coenzyme, which is the NADPH that was mentioned before, and how important that is, how that is used to reduce substrates, and how like glutathione can be reduced and then help with the with balancing your body in being not in such great oxidative stress, because all these chemicals are like signaling kind of channels that tell the body, okay, go here because this is under stress, or go here because this is under stress. And then you have to get it back to not being stressed. And that's as simply as I can explain what goes on in your body. And sometimes things are introduced to a body that really gives it on an overload. I'm surprised that how well our body does this. And being deficient, I am very surprised how well the body still compensates when you are deficient. So that's why a lot of people don't get symptoms. But there are times, especially when you're introduced to drugs and so on, certain triggers, they call them, this will cause an imbalance. That was explained earlier by Anwar. I think the best thing to realize with all of this is that, so if you are G6PD deficient, you will be deficient in NADPH, as well as reduced glutathione and many other important compounds produced by pentose phosphate pathway. And it's like a trickling effect. And sometimes you can just even have low hemolyzing going on, which means everybody has hemolyzing going on in their body, whether you are deficient or not, but it's at the rate and how quickly you hemolyze, and when you are deficient, you can be hemolyzing a lot more than a person who isn't deficient, but not to the point of where you are actually showing signs. I hope that makes sense, but that's pretty much what goes on in your body that been explained to me. I think it's interesting to know also, I'm looking at my notes here, that NADPH is essential for both, not only the antioxidant symptoms, like using, getting the body to compensate for overstress situations, oxidative stress, but it is also involved in the oxidant aspect of cells. It does have a mission of where it will create oxidation and not reducing. It's interesting that NADPH is really signaling for two things. It keeps both things going on, because sometimes we need to be in that state. Like interesting is that the NADPH is also involved in the pathway to produce reactive oxidant species, which help the cell immune systems to start a phagocyte attack when your white blood cells are invading pathogens. So although required NADPH for these types of productions can be produced by other pathways, not only by the PPP, it may require the NADPH from PPP to compensate immunity for serious infections. So this is like, it helps, especially knowing that 60% of NADPH is produced by PPP. So in other words, this is not the only pathway that produces NADPH, but it is important to help. And I think that's the points I just wanted to make out with that. So you can think about that as we're talking about screening. Why is screening so, let's see, I'm sorry, I think I got the, yeah, I think. So I guess we could go to the, what slide is that? Is that the second slide? No, not that one. Not, please, don't go to that one. Go, stay there, please. I'm sorry, I wasn't clear. So screening is really important. And when someone has G6PD, it's introduced to severe oxidative stressors. It could cause a reaction that the body is not capable of correcting. So much can go wrong. And like I said before, it's amazing how our bodies can adjust. It just being deficient with this enzyme, it makes it harder. So let's talk about the blood cells, which Anwar had also brought up, that these red blood cells, at least the mature red blood cells, are denucleated. So they solely depend on this pathway to keep them in check. And because sometimes from over-oxidative stress, it causes the actual blood cells to hemolyze or break down. And erythrocytes cannot produce enough NADPH because they are not nucleated. So that's why they are dependent on this. Okay, so I think it's important knowing that if you are G6PD deficient, you can prevent the potential attacks of acute hemolytic anemia by knowing what triggers to stay away from. Also, it was brought up about the anti-malarial drugs. There are known contraindications for G6PD deficiency. And before taking one, should definitely be tested for this deficiency. It has been reported that when G6PD is deficient in the body, it can induce methahemoglobinemia. This event is when the best way to explain it is when your red blood cells that carry oxygen by iron, and how that iron connects and holds that oxygen is what state it's in, just like these different states of being reduced or oxidized. And I think it's an Fe2 or iron 2 state where it will hook up to the oxygen. But with being deficient with this enzyme, it sometimes allows these states to be locked into a iron or an Fe3 state where it will not touch an oxygen. You'll have oxygen all over the place, but it won't take it and transfer it to other tissues and so on that need the oxygen. So that's when somebody could be in the state of called I think it's methahemoglobinemia. And what's interesting about this is, okay, so this can happen because you're deficient, or it could happen for other reasons as well. A lot of drugs, different types of drugs can also trigger it. But if you are deficient and this happens to you, the best treatment, or the most known treatment, I should say, is this product called methylene blue. And that is a severe contraindication for someone who has deficiency, and they will start hemolyzing if they are introduced to this product. So these are reasons why we should be screened, because there are times in our lives things happen, and it's better to know that we have this deficiency or not before a drug is introduced to us or any other triggers out there. Let's see. This is a very interesting thing, too, of why we should be screened. In cases of G6PD deficiency, the lack of oxidative metabolism can cause a reduction in oxygen-dependent phagocytosis. That's when I was talking about when your white blood cells start attacking anything foreign. And this allows viral and bacterial replication. This has been known when you are deficient. The type of disease that's been documented a lot was, I hope I say it right, granulomatous disease. Also, research has found that the occurrence of neonatal sepsis is significantly increased when an infant has severe G6PD deficiency. So infections are known to cause oxidative stress with those who have G6PD deficiency, and have been reported, hemolysis have been reported. It's kind of interesting. We were talking just previously about the anti-malarial drug HCQ, or hydroxychloroquine. And it is a known treatment for COVID, as it was brought up before. Has a hemolytic episodes of those have been reported of those infected with COVID and have G6PD deficiency. So what does that mean? There have been other reports out there saying HCQ is fine for those who are G6PD deficient. There was a wonderful report about I shouldn't say wonderful, it was a very strong report. I think it was over 300 and somewhat patients that were doing research for lupus, and how they found that HCQ is a wonderful drug for those who have lupus. And in this study, they also studied for, they found that part of the population was G6PD deficient. And it's interesting that this report then kind of said in the end that it seemed to have no effect of anybody who was deficient. The problem is, again, like I said before, is that if you heard what I actually meant was that there have been reports with COVID, which is a virus, which is an infection, that when they use this drug, HCQ, there have been hemolytic episodes. So in other words, maybe the people, because they were not sick in that study, did not show any signs of having any contraindication to that drug. But if someone is sick, it seems to be that people are showing how these drugs can really affect or trigger hemolysis when somebody is deficient. I hope I'm clear about that, and maybe you can ask questions later. Now you could show picture 3, please. This is something that is very, very dear to my heart. This is a slide of predictors. I always get upset when I see this because this could be avoided. Now, newborns that have G6PD deficiency are vulnerable to severe neonatal jaundice. Doesn't mean that they're going to have this happen, but they are more vulnerable, and which can lead to kernicterus. This kernicterus is a type of brain damage that is done really on the motor skill area of the brain. What happens is a lot of newborns who are G6PD deficient tend to they have like a 30% chance of building into a hyperbilirubinemia state. What does that mean? Well, that means that when your red blood cells start breaking down, they produce a toxin called bilirubin. Bilirubin, when if bilirubin, enough of it is happening in a cell, that's what actually causes the cell. It's a toxin and the cell will break down. That's really what hemolysis is. And now this bilirubin is floating around in your body, in your bloodstream, and you become yellow. Is really a good sign of jaundice. There are other signs as well. Now, when we talk about a newborn and they have hyperbilirubinemia, that means that there's such a large amount of bilirubin. And this state is very, very dangerous. The problem is that this doesn't happen all the time, but it does happen, like I said, 30% of the time. I know in the United States, I think it's close to 80% of the babies born are jaundice. Does this mean they have G6PD deficiency? Does this mean they're going to go into a hyperbilirubinemia state? No, that does not mean that. There's something that is causing these children to be jaundice. It is a normal, they call it a physiological state of jaundice. And it goes away and everything's fine. So I think that I feel that there is like a a little laid back when a baby is jaundiced, because it is so common that not knowing they are G6PD deficient allows things like kernicterus to happen. So I truly believe that that newborn screening for G6PD deficiency is so important. So things like this do not happen, because they will look at that jaundice state a little closer. They have a certain protocol that kind of tells them that things are going the wrong direction than they would if with a normal person having a normal baby being jaundiced. So it's interesting that, I'm sorry, I got a little upset, but as you can see, a lot of these babies do grow up, but they mainly cannot walk. They have hearing issues. They can't speak a lot of times. They are usually, unless it was such a severe state of kernicterus, are cognitively perfect, intelligent. They just have no way of communicating. Many of them use things like they use laptops, to communicate and things like that, or to point to things and so on. They are very strong people, but they have very, very strong disabilities as well in movement. And also sometimes they usually end up with cerebral palsy. There are many reasons for cerebral palsy. Kernicterus is one of them. I also wanted to state that kernicterus is not only caused by G6PD deficiency. There are other things out there that causes this, that will cause the brain damage, that will cause the high hyperbilirubinemia to occur. And a lot of times, the reason why this does slip by a lot is that they have found that babies that are born and are G6PD deficient are at home when this hyperbilirubinemia activity occurs. Thus, there isn't somebody looking at them or taking their blood type, their blood test. Usually they're home, and this usually happens by day 3. And it can happen 2 weeks later. It's all according. Maybe the child was introduced to a trigger, and triggers can be passed on through the milk of the mother as well. And the mother not knowing about anything about G6PD deficiency or knowing that their child has it and so on, it can really cause something like this to happen. So I think that I strongly feel that this is something that a very strong reason why everybody should know. If you're planning to have a family, you should be screened for G6PD deficiency as well.
Keely, I want to add a sunny side to the story. And Keely, maybe you might need to mute.
Apologize.
No problem. So and I see these pictures and I feel so strongly for Keely and for folks who have, unfortunately, have to go through life like this. I have a sitting example of the other side of the story. My son was born in India, and his doctor was very well aware of G6PD deficiency and has been championing neonatal tests in India to make that a mandatory sort of option. So I was very fortunate to have him as my doctor. I personally know about G6PD. I knew I was deficient because my dad reacted badly to medication. And so he had got the test done. So going into my pregnancy, I was very clear that I was deficient. And if it was a son, we were sure that he would be deficient. And we were able to take care. And actually, Keely, you said that people are sent home. I remember my gynac coming into my room, I think 2 days or 3 days after the delivery. And he was like, "What are you doing? You're still here." And that's, if I had a doctor that was not aware and was not treating my son for jaundice because of G6PD, we might have been home. And if I was not aware of G6PD either, my son might have been jaundiced at home without knowing the implications of what that could have been. So I cannot tell you how fortunate I feel because I have a very healthy, happy child. And I cannot tell you how fortunate I feel that, one, I knew that I was deficient. And I do come from a family of doctors. So, my mom made sure that we check, and my mother-in-law is also a doctor. So everyone was very, more aware. And of course, my doctor itself, my neonatologist, was very well aware and was championing this cause. So, and it still is championing the cause. And we did do the newborn screening test as soon as he was born. And of course, he came back deficient and he had jaundice. And he had to stay in the hospital for, I think we stayed there for at least 5 or 6 days, which is not normal. Like I said, usually people are sent home in 2 days itself. So, it is just so important that these things don't have to happen. And this can change. And that's the reason why this topic is such an important topic at the neonatal level going forward. And also now during COVID, because there's so much that we can change in terms of treatment options and how G6PD individuals are addressed. I'll mute.
And so towards the end of this, we'll talk about how people can get tested, correct?
Yes, these are important points to really understand why one person should be screened and tested. Before you discuss the type of tests and so on, this is so important to understand. This is just not, you just don't go and get tested. There are reasons why you should. I think it's also very, very important to understand the genetics of the enzyme before being tested, because it really, understanding the genetics aspects of it will help you, and that's slide number 4, will help you really understand what type of test is most accurate for you.
So do you want me to advance the presentation now?
Please, to 4.
All right.
Okay, so let's understand about G6PD enzyme. First of all, I think you got to really understand, I think the first thing you should really understand is what is a chromosome. A chromosome carries all your genetic information. This is the key to who you are. It really is. So, and everybody is different. But there are some things that are similar or alike. And I think you should understand that you're carrying all your genetic information. This picture up on the top, it will show you 22 pairs of autosomal chromosomes. They're very important. One pair is from your, one of the pair is from your mother, and the other pair is from your father. Simple as that. Then there is this special pair called the sex chromosomes, and that is the one circled in red. And this one is particularly showing you being a male. You have an X and a Y. You could also have 2 X's, and that would determine you being a female. I think it's important to understand that that there are, why are we referring to the sex chromosome is because this enzyme is located on the X chromosome. So what does that mean? Okay, so all the genetic information of that enzyme is located on actually the lower arm of the X chromosome, which is known as the Q area. There's a short arm and there's a long arm. But also, this brings into interest of that about the X chromosome in itself and the gene located for G6PD deficiency. That area is the most polymorphic loci or locus in humans. It has over 400 variants. Just that area of where that gene is located, there's 400 variants, known variants that they know of, and about 180 of them mutations have been found. so far. So we have 180 mutations found. And what does that mean? That mutation, that 180 mutations, I'm sure there's a lot more, but that one deals with the deficiency aspect of G6PD enzyme. I hope that makes sense. So let's see. So that determines the effect of the deficiency of the enzyme is exactly which mutation that is. That is also known as words you may hear variants. There's 180 variants. You may hear that term as well. And these variants have been classified by the World Health Organization by the residual enzymatic activity of it. So they classify it as 1, 2, 3, 4, 5. I am not going to go into that because that's not as important right now for screening, but I really think it's something that you should look into, and it is being changed. They are addressing that issue in their classification as well, the World Health Organization. I think you should know that the most prevalent in USA the most prevalent variations are African and Mediterranean variant. And there are reasons why I'm telling you that as well. Let's see, usually it is brought down, these mutations are brought down, found on the X chromosome of in this gene area, is brought down within the family. It is not, because it's carried on the X chromosome. Very rarely is it a spontaneous mutation that happens, but sometimes it can. Actually, because it's so polymorphic, it could happen, and it has happened. So about 99% of the time, it's brought down. So if you have it, that usually means someone else in your family has it. So to understand the results of these tests, one should understand of what this all means genetically, having the deficiency. And that's what the right side of this slide showing you, that you can have an X, whether it's deficient or not of this enzyme. It has that mutation. And female has 2 X's, and male only has 1. Because the male only has 1, either that male is deficient or he is not deficient. It can't be in between. And that is called a hemizygous, is the term they use for that. A female, as you know, has 2 X's. Interestingly enough, the female could have only 1 X with a deficiency or both X's with a deficiency. And each X could have a different variant of the deficiency. So it can get very complicated. And that's really when you go see a specialist in regards to about genetics. You really got to see a counselor, and they can explain this clearer. This is just showing you the diagram. The reason why I'm showing you this is so you have I would like to talk about the female. If the female has both X's that are deficient, she is known as a homozygous deficiency, of the G6PD deficiency, which is exactly like a male. All the X's are deficient. Thus, that person has the deficiency and expresses it as such. Now, let's talk about the heterozygous female. This is where it becomes interesting. And this is probably there are more heterozygous females with this enzyme deficiency than not. And than being homosexual, genetics, homozygous, having both. What happens here is that you would think it would be 50-50. So, okay, 1 X has it, 1 X doesn't. So 50% chance, I really have nothing really to worry about because one will compensate for the other. And that is a very old way of thinking. There is this term called X inactivation, or known as lionization. Every cell in your body only needs 1 X. The other X gets turned off. It doesn't be expressed at all. It's interesting that, well, which X will this person have in one cell? Will it be the X that is deficient that's on, or the X that isn't? And that determination is still being done in research about the X inactivation process, but it does happen. So what I'm trying to say is when you are heterozygous, you will express the deficiency by the amount of cells that have been, of the X cells, that X chromosomes have been turned on that are showing, that are expressing the deficiency. So what does that mean? Like, you really don't know how badly you are deficient in just looking at your chromosomes. You wouldn't know. Okay, so that's why I brought this up. I hope I was clear about that. So that's why the accuracy of test results for each type of test is based on sex chromosomes of the patient. And what does that mean? I'll explain to you soon. So I think we are ready to really talk about screening. So we could go to slide 5.
Keely, can I just, for the audience, the non-scientists out there, Keely just used a couple of genetics related terms. So we're really in the realm. Can you mute for a second, Keely? Just for 1 second. Yeah, so this is all in the realm of genetics, which she's showing you. Those are called Punnett squares. I haven't seen those since undergrad. So what you're doing is you're just predicting, you're looking at the likelihood of what the child is going to inherit based upon, I believe it's the alleles that the parents are carrying. So you really have to understand that the terms homozygous and heterozygous, homo meaning the same and hetero meaning different. So you're usually, if you look at those tables, a woman can have the allele for, geez, I haven't studied up on my genetics in a while, but she has the allele for the deficiency and the non-deficiency. So she can have the deficiency on one of her X's, and she can have a normal X, or she can carry that would be heterozygous, and she could have the deficiency on both of her X's. That's homozygous, but she could also have 2 normal X's, which is homozygous as well. So you're thinking about same versus different, and you can have a mixture of the two. But for a man, he just needs that one X to have the deficiency, to have the phenotype. So I just wanted to add that in for the non-scientists out there.
Thank you. Sometimes I carry on and I need to come down a little. So okay. I apologize.
So we're gonna go to the next slide.
Yeah, because I don't know if you want me to go into if a father is deficient, he will only bring that down to his daughters and all his daughters. So looking at these Punnett squares would tell you that. I could tell you something else. If the mother is deficient, it's according whether she's homozygous, she has both X's with it, she will definitely make all her sons will be deficient. If she's heterozygous, she has a 50% chance of bringing that down to her son. And that's the same with her daughter, in the sense that if she's homozygous, she has a 50% chance of giving it to her daughter. If she is heterozygous, she still has a 50% chance. She has 100%. She has a 100% bringing it down to her daughter, but her daughter, it's according what the father is, whether or not she is homozygous or heterozygous. If the mother is heterozygous, she has a 50% chance of giving it that one allele to her daughter, and like I said, and 50% to her son as well. So it's all according, and this is something really you should see a genetic counselor about if you find out you are deficient and you want to have a family. It's something to discuss.
Okay, I'm gonna bring those next slides up. I know Terry and Giovanni are waiting patiently backstage.
Yes, I know.
Niloufer wants to say something. Go ahead, Niloufer.
Yeah, I just want to add, finding out that one person in the family is deficient, and Kili, I think you might have to mute again. So it's even finding out one person in the family is deficient can significantly help your entire family and background, because and your extended family. And that's exactly what happened to me, right? Only because we found out that my dad was deficient, that we realized that we could be deficient. At that time, I didn't know heterozygous and homozygous as well. And we got tested. And so it is so important, even if we start testing like the neonatal testing that Keely is advocating for. If the families find out that one person is deficient, they can extrapolate that and try and find out, how many others in the family or the extended family could be deficient. And that can also help with treatment options and with how we manage, of course, during COVID, how we manage the pandemic, but also post-COVID, how you look at more personalized medicine and more, I think you said pharmacogenetics, it can play a huge role there. So it's very important that we start testing individuals for this deficiency and get much better at addressing the concerns when they are deficient. Keely, I'll pause there.
Thanks. That's very well pointed out, because it is so important. Once somebody in the family has it, it may help the entire family's health. So let's go to I think we're finally ready to talk about the types of screening, because I think you've got a good idea now of about G6PD gene and how it works and how important it is. So there are really two types of tests. There's a phenotypic test. It measures the G6PD enzyme activity in blood samples. And then there is a genotypic test. And this is a detection of mutated gene by DNA coding, either by saliva or blood samples. So what does this mean? There are different reasons of why you would do different tests. So let's look at the phenotypic tests. And the World Health Organization has this pre-qualification technical specification for diagnostics to measure G6PD activity, because that's what these phenotypic tests do. They measure the activity of the G6PD deficiency gene. So why are all these different tests? You would think it would be pretty easy. Well, the most simple test is called a qualitative test. A qualitative test is like a yes or no. Either you have it or you don't. But what does that mean? It's really when it's less than 30% deficiency, I should say G6PD activity. The actual enzyme activity is being measured. And if it's less than 30%, it's yes, positive for G6PD deficiency. If it's greater than that, no. And the interesting part about that is it's missing a lot of people, because it is missing the people that are between 30 and 80% deficient. Those are usually the people that are heterozygous, and are females heterozygous. They have a mix. They are part deficient. One X is, one X isn't. And those X's, how they're expressed, explains the different activity of the actual enzyme. So that is why the WHO also states that it has to have the ability to discriminate from normal and being deficient, as they say, which is less than 30%, and intermediate. Now, don't get the idea that because you're intermediate, you're not deficient. You are deficient. You are just not severely deficient. Or, there is you could be expressing the deficiency quite a lot. It could be, but they use the term G6PD deficient as those who are less than 30%, which is usually a homozygous female. And it also includes, of course, the hemizygous male, the male that only has the X and it's deficient, and a female that has both X's that are carrying the deficient gene. Okay, so then they have a semi-quantitative. What's good about, I should tell you the good things about qualitative tests. It's great, like I said, for hemizygous males and homozygous females. It's inexpensive. It's good for like point of care uses. In other words, you don't need a facility. Usually you don't need a facility. And it cannot, and the bad thing, like I said, was when I explained that it doesn't talk at all about it doesn't explain heterozygous or even determine if they're deficient if you're a heterozygous female. Okay, now they have a semi-quantitative. This is interesting because semi-quantitative, it's really a qualitative test, but it does a little more. And what this does, it still only presents accuracy for less than 30%. So it's still eliminating those that are heterozygous females. It is not, they can't interpret. But it is these different ones that are semi-quantitative are used a lot, especially in areas where they have to be in the field, or or a point of care right there and not have a facility. Or some of these set up where you can have a little camp, and they'll have things there under their tent, but they per se don't need a big institution to be doing the test or interpreting the test. These semi-quantitative tests, I know you've heard of them if you've looked at any of the literature of screening, like the fluorescent spot test, the FST it's referred to, the colorimetric assays, which is the MRT, and then there's a WSTA, and they look at color. Actually, the color will determine what's going on, whether you're deficient or not. If you show color, usually you are not deficient. If you show any kind of color, you are deficient. And I hate to say it, but it still does not, it still may give you some indication if you're heterozygous, but there's a lot of females that this would not show this would show color. And that would be interpreted as you not having it. So there's also the lateral flow test, which is also very important because that's being used a lot too. And there are issues with that as well. This particular lateral flow test really is important. A lot of these, I should say, semi-quantitative tests do not take the blood in whole. And usually if somebody hematocrit is off balance, in other words, the amount of red blood cells versus the blood in total is off, it can really skew the results. If your hematocrit level is low, sometimes it will give you a false negative. In other words, it would not pick up that you're deficient. And if they give you a lot, if you have too much red blood cells in that sample, it would give you a false positive. It would tell you that you're fine and you are not. So these tests are very tricky. So you cannot say it's science. You cannot say, yes, I definitely am, or I'm definitely not. These two types of tests. So then there's the quantitative test. This particular test is very, very interesting because it actually reads out a an amount of the enzyme activity. It will be giving you an actual amount. It's not really interpreting. It just gives you this number. And it will show those. It's very good for those that are less than 30%. And it's also very good for those that are not deficient, which is usually greater than 80. But it's that in between it's not as accurate all the time. And that is a concern because it doesn't, the accuracy of that is usually probably for that population, the heterozygous female population, has been found to be between 50 up to maybe 70%. It will pick it up and be accurate. So that's a concern. So when I talk about these quantitative tests, they're like using a photometric assay, using a spectrometer. You actually need a facility for that. So a lot of hospitals have these, and so you could send numerous amount of samples to a hospital to test this. Let's see, what else can I say about this? The semi-quantitative, quantitative, cytochemical assay seems to be the best way to determine the heterozygous female population. So you go, aha, that's it. That's the answer. We can use it. The problem with this, it's mainly only used for research, this type of procedure. And it does provide information of the intracellular G6PD level, of their activity level. It's great, but it is not good to use for, it's too costly, the test takes too long, turnaround, compared to the other ones, than if you were out in the field and you needed to screen somebody for this. They found that this type of cytochemical assay, they want to work on it to simplify it so that it will be much easier for a female, heterozygous female, to be confirmed that they are that they had this deficiency. So on that note, there is now the genotype test. Now, as you learned before, this will detect mutated genes by DNA coding. And DNA sequences, there are two types. And this is something that's important to know. DNA sequencing is they will take the entire area of that of the G6PD gene and screen and sequence it for any kind of mutation that they know, and that's only 180. There are other mutations out there, and that is not being picked up this way. But it's the best way to make sure you got at least 180 variants. You have a very good chance of it picking up and being diagnosed. With a snippet, they call it, or a PCR-SNP analysis, is they only pick certain variants, and they're screening for those certain variants only, not the entire spectrum. And what's interesting about that is like 23andMe, wonderful, wonderful company. They are screening for 2 types of variants, and that's all. So if you get screened by 23andMe and it does not state that you are G6PD deficient, does not mean you are not G6PD deficient. It's just that they only screened you for the Mediterranean variant as well as the African variant, because that's the most prevalent variant in the United States. So on that note, there are complications of these tests, and the complications of these tests, I guess we could explain the next slide very quickly.
So Keely, before I go on, just in summary, is there one test that you think is most effective over the others?
It's according why you're screening. If you're screening to see if you are deficient for malarial reasons, so you need to take the drug, let's say that. A semi-quantitative test will quickly tell those, except for the females that are heterozygous. And they have to be educated enough to know that when they're being tested, you know, that part of that population, some of these semi-quantitatives are pretty good. They'll catch a lot of them, but not catch all of them. And same with the quantitative. They'll catch some of them, but not a lot of them. So if you're out in the field, usually semi-quantitative is really the way to go to try to catch as many as you can. It's not perfect, and that has to be known. So I'm sure Tavani will explain that more when she talks about it, about the screening for that. Now let's say you're having a family and you want to know if you have this or not. Probably you could start off, it's according to cost, because DNA sequencing is costly. I don't know if your insurance will cover it or not, but that would definitely tell you, at least out of the 180 variants, that you have this. If you know that you have the background of being Mediterranean or possibly being African, then maybe you can start off with the PCR and using 23andMe or a company like that. And that would at least eliminate, that would probably be your first point of starting to get tested to see if you have this. Quantitative is very cheap. It's just not as reliable as genome when it comes to something like that. But it does catch a lot. And then the cytochemical assay, they're getting to the point now where they actually take the semi-quantitative tests, like the MET test, which is a methemoglobin test, and then they bring it over and they actually create a cytochemical assay afterwards, and it shows up beautifully. It's just a little time consuming. It's not that expensive. It can maybe be about $20 to have it run per se. I don't know if they would charge you, I have no idea, but to actually run it, it's about $20. So that may be the way to go, is to have a semi-quantitative MET test and have it go into the cytochemical assay, where you're using a cytometer to determine it. And it shows you peaks. It will show you 2 peaks if you are heterozygous female. It'll show you 1 peak of normal and 1 peak that's not. And it's really pretty explicit. So that's why I could go on and on and on about this. I will never tell you the best. So
okay, so for the audience, Keely's described numerous testing methodologies. In science, different tests have different advantages. They have disadvantages. Some are more effective, some are more quantitative, some are more qualitative. And there are different detection methods. So she talked about PCR, which is testing your DNA. And then she just mentioned flow cytometry, which I believe is testing cell types. So I just wanted to just give them a feel for what tests will be more effective and which ones aren't. But so shall we go to the your final slide? Okay. I'm going to step away for a second, but go ahead, Keely. So
I'm almost done here. So complications with these tests. Besides what I told you, there are more complications. And these complications are pretty important. Hemolyzing with skew phenotypic test results. In other words, if you are hemolyzing, you should not do any of those quantitative, qualitative, any of those type of tests, because they will be skewed and you'll get false negatives for it. The influx of G6PD enzyme found in immature blood cells are known to change the reading of the personal actual amount of this gene if it's present. So one should wait about 3 to 4 weeks after hemolyzing before getting tested. This is something you discuss with your doctors. Qualitative test results can be skewed by abnormal hematocrit levels in samples for certain tests, like I explained for that lateral flow semi-quantitative test. They're used a lot, but if the person doesn't have a normal amount of red blood cells, you will get the wrong reading, like I explained before. Qualitative and semi-qualitative tests can only detect, as you know, heterozygous male and homozygous females. Quantitative tests vary in the sensitivity of detecting G6PD deficiency in heterogeneous females. This definitely needs room. Genome tests, they tell you they do not have anything to tell you about the quantitative measure. So even if you know you genetically by a genetic testing that you are deficient, it may be a good reason for you to go and find out how deficient are you. Not only the variant, but also how, it does tell you the variant, but it does not tell you how deficient you are of that variant because of that variant. DNA sequence analysis give false negatives for any new mutations not yet coded, which I explained, and I also explained how the snippet was limited with their panel of what they're screening. So you may be missed out because your variant was not on that panel. So that is really it. I really wanted to explain what we're doing as G6PD Deficiency Foundation. I'm proud to say that we are right now working on, with an advocacy company, that Millennium Strategies, to get the New York State to pass a bill that will pass a bill for newborn screening for G6PD deficiency. If this is going on right now, I'd appreciate letters of support. If you want to send them to me, you can at kharris@g6pddeficiency.org, or you can talk to your legislators if you are in New York and tell them that this is something that is very important, that newborns need to be screened for this. I'm very excited about it. And then we will go forward from that. Either we go state by state or we're going to go nationwide. So this is something, and we're going to be pushing it worldwide as well. I think it's going to be a domino effect, but a very positive one.
So, Keely, you mentioned New York State. The people in the audience, they live in different states. Is this something that is being done at the discretion of every individual state, or is there a uniform policy? Can you talk about that a little bit?
Sad to say, there is not one state in the United States that has a newborn screening on their panel. It's not mandatory, and it should be. And in that note, there are areas in certain states that screen automatically no matter what, because they believe their population has a high prevalence of it, such as Washington, D.C., and I know Pennsylvania, also a lot of areas like Philadelphia and so on will screen for it. That's not the way it should work. It should be universal. And the United States has a universal newborn screening program. And that is where we're going to be going to get it pushed so that this guideline will be followed by all the states. So right now, hopefully, New York. And then we'll go from there. But it's a lot of work. It's a lot of work and a lot of awareness. And that's what's really important about this, is the awareness, so you have the public behind you to push this to get it done.
Okay, well, so I have a question here from Herbert Lewis. He says, can she say that again so we can put it in the chat? I'm not sure if he's referring to your email, because you stated people can email you. Or, yes? Yeah,
people can email me if you want to write a letter of support. You do not have to be from New York. United States would be great, but it doesn't matter. It could be worldwide. And my email again is k.harris@g6pddf.org. And you can find that also on the website.
K.harris@G6PDDF. So 2D.
.org. So I also want to add, in terms of screening, I think, yeah, thank you. I don't know why it's your computer, but the fact is that in terms of countries, that not just the states and US, but worldwide, there is a lack of awareness. There's a lack of awareness. I know Tivani can speak to it. We have another doctor from Senegal who's on our task force, and he speaks to it all the time. There is, even in the highly dense populations where the prevalence is really high, there is a very, very poor understanding and awareness of the deficiency. There are a few countries like Singapore that do neonatal screening, and you will see, even if you look at how COVID has been managed, and I'm not saying it's a direct correlation, but it's interesting to see that countries like Singapore have been able to manage the pandemic as well much more effectively. And they do neonatal screening. And I think there's so much to learn from the rest of the world, and just so much for everyone to be able to support each other and the different countries to come together and really make this a global topic. I think more than, it's definitely relevant in the US, it's relevant everywhere around the globe. And people need to be aware, especially in a pandemic like COVID.
Okay. So I want to thank Keely for that presentation. Keely gave us a lot of information. I was thinking just now that I might shoot some follow-up videos just on the testing methodologies that Keely talked about, so you all can have a better idea of what PCR is and what flow cytometry are. But I think the important thing is to have these bits of information that you can at least ask your physician about. And I think, yeah, you can watch this on the playback because there was a lot there. But the important thing is just having the information to ask your physician about. And Keely gave us a lot of valuable information there. So with that, shall we invite up Terry and Tivani Nilufer? All right. So we have two more guests backstage, one from South Africa. So let me stop this. And this will start the second part of this discussion. And we'll bring up Terry. You all have been very patient. And Tivani from South Africa.
Hi. Hello.
Hi,
everybody. Yes, thank you, Tilly, for that background, for that detailed background on this, G6PD deficiency and your perspective from the US. And although, that's what you were saying was from the US perspective, and it's probably the same all over the world. And I liked your, towards the end, talked about the testing, which is the area that I obviously work on. And I was very much interested in that presentation of the different tests and what's available and what's reliable or not reliable. So I'm just going to share my experience on diagnostics, on G6PD diagnostics, and my experience on that. So I got exposed to G6PD deficiency, and I mean, I'm not an expert, I'm not a hematology expert, but I'm a medical scientist, also trained in hematology, and also did a lot of diagnostic tests in the hematology. And one of the tests that interested me a lot when I was working in St. George's Hospital in London, in the hematology lab there, it was that we screened G6PD for all malaria tests. They all had to G6PD before, clinical decision. And I started reading a little bit more about it, and obviously most of the people.
Oh, that's so unfortunate. I think we just lost Tivani.
Okay, I was ask, wasn't Terry supposed to go first? Or
either way, I think we can. What I can do is I can introduce Tivani while she's coming back, and then we can, and Terry as well, and then we can go from there. What do you think, Anwar? While we wait for Tivani to
back.
come back.
Yeah, why don't you introduce her first?
Sure.
And then, all right.
Yeah. So I know we jumped right into it, but I must say Tivani has a very impressive biodata. She is Dr. Tivani is a professor and deputy dean of research and postgraduate studies for the Faculty of Health Sciences in South Africa. She's an NRF-rated medical scientist there. And she's authored more than 100 peer-reviewed publications in journals including Lancet and Nature. She's also a member of the COVID Scientific Advisory Committee for the Limpopo province, and she's one of the co-investigators for national COVID study aimed at optimizing implementation of point-of-care systems in South Africa. And her research work on point-of-care diagnostics has been recognized nationally and internationally. She's won multiple competitive research awards. She was listed in the Mail and Guardian as one of the most powerful women in 2020. And she's been invited to join the University College of London in collaboration for advancement of Sustainable Medical Innovation Fellowship. She also serves as an editorial board member for Lancet's e-Biomedicine Journal, and as an associate editor for Frontiers Reproductive Health and guest editor for the Diagnostics Journal. So I think we might just need a session exclusively with Tivani I'm sure she will have a lot to offer. And like she said, she came across G6PD when she was working in London, and she's very passionate about the topic as well. And we're very honored to have her as a part of our task force. The other guest, of course, sorry, Anwar, you were saying something?
No, Lufa, I just sent you a message in the private chat, and I think that might be a good idea. Can you see the message?
Yes, I did see that. Yes, I think if we're not able to have Tivani join back, we could probably plan another session exclusively with her. I think that would really help. And of course, in terms of Terry, Terry is one of the most passionate COVID G6PD and COVID task force members. Terry, of course, is just like me, not necessarily from a science background, but I would say is one of the most well-read folks in our task force. She's very passionate about the subject. I know Terry's had G6PD in her family as well and can talk about her experiences. But Terry also was the one who brought up and brought to our attention the fact that there's, there are studies in the US about, the military has done a lot of studies in the US about G6PD. And, there's a good opportunity for us in the US, and I hope worldwide as well. I'm sure there's so many opportunities to start looking at clinical testing to really see how G6PD impacts COVID. So.
Okay, so Terry, I have you. Let me start your presentation right here.
If you could, I'm mostly going to talk about the table. So if you want to put the table up first, and I'll talk about that, and then the other ones we'll go through briefly. But I'm going to do a lot with this. Okay. So I just have and Anwar putting that up there so people can start to just kind of think about what that is. This was released, a report was released December of 2019 by the U.S. military of G6PD deficiency in 2.3 million active duty service people. Okay, that's what this is. And if you go online and you just Google search or whatever, G6PD military, this is like probably one of the first things that will come up, is this whole study. And this is just a table in there that I'm going to discuss a little bit about. But before that, what I want to talk about is I first became aware of G6PD deficiency in 2013 when my mother had a lot of reactions suddenly. She had been healthy and not taking any medications. And in our family, we kind of had bad experiences in the past with medications. And so we just tended to just try to stay away from them for the most part. And we survived pretty much by that. But my mother had a lot of reactions, many of them similar to reactions people are having today with COVID-19. And so I went on a journey to try to figure out what was going on, and that's how I came across G6PD deficiency for the first time. And there have been people who were not. There's a case study recently that you can also look up online where a man was 86 before he was discovered that he had G6PD deficiency, and I think that was in a case of methemoglobinemia, like Keely was talking about earlier. So even though fauvism is a term like for people that have a reaction to fava beans, and that's been known. For centuries, like in the Middle East, people have known to not that they may have a reaction to fava beans or to the henna dye, things like that. Okay. But it really was the US military that first discovered more about G6PD deficiency and about the genetics. Okay. And why was that? It had to do with malaria medications, as was mentioned. And they were testing Pamaquin first on prisoners, and then they were given Primaquin to soldiers, and they were finding out that the Black soldiers, some of them anyway, were getting severely ill. I believe some actually died. I do remember reading that in 2013 when I was researching it, but in looking for it now, I haven't been able to find that. So this is where they first, started studying, like, why would this be? And that's when they first discovered some of the genetics about it. Now, something important to remember about genetics is there's a lot of things in genetics that can be advantageous under certain circumstances and disadvantageous under other circumstances. Like, I myself don't even really like the fact that it's called G6PD deficiency, and it makes it seem like it's a terrible illness or something, okay? The fact is that in a totally natural society or totally natural surroundings, probably people with G6PD deficiency would be some of the healthiest people around, okay? Because it does protect against severe reactions to malaria, as well as certain other pathogens and cancers, okay? But the fact is, right now, in our modern world, we're surrounded all the time with all kinds of oxidant stressors, from pollution, different chemicals and pesticides, a lot of the pharmaceuticals, even EMFs and radiation, things like that. These are all oxidant stressors, and depending on our level of G6PD deficiency and depending on the amount of the oxidant stressors and the combination of them, could be the difference between whether we have bad reactions or not. Okay, we may not always have a bad reaction to a certain thing, depending on the dosage of it or the level of it or the combination with others. Okay, And it's also similar, again, like I said, the advantage and the disadvantage, it's like skin pigmentation, okay? In a area where there's not a lot of sun and you're not exposed to much sun, lighter skin is fine, maybe even an advantage. It absorbs vitamin D more, okay? But in another situation where there's plenty of sun exposure, darker skin is more advantageous and less likely to get cancers and things like that. So I just want to move away from, like, it seems like there's been some people that don't want to look at and study G6PD deficiency because they look at it as a, quote, deficiency. And, oh, I don't want to be told I'm deficient, or, you know, my group of people is more deficient in some way or whatever. And that's why I think it's important to look at it instead as advantages and disadvantages, okay? So the, you know, as I mentioned, the military first discovered this kind of the sciences behind it and everything, and about that it was a problem with the malaria medications. And Black Panthers picked up on that. And the Black Panthers were really our last big major advocates for people with G6PD deficiency. If you look at some of their old newspapers from the late 60s and the early 70s, they had a lot about it. And they set up their health, they set up their clinics, and they did screenings for both sickle cell anemia and G6PD deficiency. Now, they didn't get it right on everything because at that time, they thought females could only be carriers. They didn't know that it could affect females, but they did make it a big point of their health clinics and their health screenings and their education. So if they had continued to really survive, I mean, I know there's some that are still around, but if they had continued as an organization, I'm sure we would be a lot more advanced on the public knowing more about G6PD deficiency. But unfortunately, that has not happened. And right now, 95% of people with G6PD deficiency are undiagnosed. And even those who are diagnosed don't know a whole lot about it for the most part, and their doctors don't seem to know a whole lot about it. And the Black Panthers, another important thing, if you look back at some of their newspapers, they warned that they were afraid, it seemed like, or maybe afraid isn't the right word, but they kind of forewarned that this advantage could possibly be turned to a disadvantage by people of ill will, that could be a possibility to watch out for, okay? So anyway, just something to think about. So now we go on this to the table here. And again, this study was done between 2004 and 2018. It was released last December of 2019, okay, a little over a year ago, okay? So let's take a look at it and see the non-Hispanic white males, 0.4% in the military of active duty service people have G6PD deficiency, okay? And 0.3% of the females that were in, okay? Then look at the next highest after that, since that's the lowest number. The next would be Hispanic, okay? 1.7%. So that's like 4 times the amount of the non-Hispanic white, okay, for the males. And then the next group, oh, I'm sorry, I should have gone to American Indian first. American Indian and Native, Alaska Native is 0.9%. So that's a little more than twice as much as non-Hispanic white, okay? And then Hispanic is a little more than 4 times, okay? Other and unknown, most likely would probably be like people that identify, they're so-called mixed race, as people put it, or whatever, and they don't want to pick one over the other. And so that's also quite, compared to the non-Hispanic white, is quite a bit more, 6 times, okay? And then you go to Asian Pacific Islander is next, and then last, the non-Hispanic Black is 11.2% for the males and 4.7% for the females. Now, another thing, I'm not sure, I don't remember Keely going over this, but she might have, about when we talk about the different variants too. Something important to know, that the South Asian version and the Mediterranean version or variant are tend to be more severe than the African variant. Okay, so that's important to keep in mind too. So the way that they did this was they did use the cytochemical assay test that Keely went over there. So they did not do, the genetics. Now, one thing to know is that females would typically be genetically heterozygous. Females would probably be about 4 times the amount that they actually are showing here. These are the ones that actually, when they did the blood level was, was a lot less. Okay. So, but they found out, for instance, that African-American women, about 20% are heterozygous, and a little less than 5% here, 4.7% actually have lower levels in the blood. Okay. Another thing about this study is that they did look at the levels. Okay. And I think Keely went over, but I'm not sure totally, the 5 levels, where 4 is normally considered normal, quote normal. Okay. But even normal is within a range. It's not like one, one number or one amount. 5 is considered more than normal. Okay. And 1 through 3 are considered less than normal. Okay. Level 3, I'm pretty sure in this one, I know sometimes when you look at it on World Health and you look at it military, it may be slightly different. But what I'm looking at right now, level 3 is between 10 and 60% of normal. Level 2 is less than 10%, but not constantly hemolyzing. And level 1 is continuous hemolyzing. Okay, chronic. Okay. So what they found was that no active duty service members had level 1. Okay. So why would that be? I don't know. Maybe people who had level 1 did experience some, illnesses or whatever. So, I mean, your active duty service people would be the most physically fit cohort, right, in the population for the most part. So maybe people that are level 1, by the time they're, old enough to enlist in the service or whatever, they've already had some issues. I don't know. Level 2, there were not very many level 2 either, and most of them were Hispanic and the Asian Pacific Islanders that were in the military. Okay, so most of them overall were level 3. Okay, so anyway, the point with this is the military knows a lot about G6PD. They know the status of every active duty service person, the G6PD status and their level. Whether they always put it in their health records, whether the people understand or know that, is a question, because I've talked to some who really didn't know. All they know is they can't take the same malaria drug that other people do, or they might have been told, something about it, but not that much unless they are more of the officer level, where they have healthcare professionals in their family or something like that. and it looks like the military has gone through kind of phases where they were saying that people without with G6PD deficiency couldn't take any malaria meds and also could not go to malaria areas that had malaria, and then they switched and they've changed back and forth. So I'm not sure exactly what the protocol right now for that is. But the as I mentioned, there's reasons why people, especially those who are undiagnosed, which 95% of people with G6PD deficiency are, as I mentioned, if they're undiagnosed and they don't know, the doctor doesn't know, things are likely to happen that are not, can I say, caught, or like a lot of times, I mean, doctors are looking at so many different things. They don't, they don't always know like what to look for because there's so much. But I'm just saying, a lot of the reactions that people have had, like with certain inflammation all over the body, with people having sudden heart problems or embolisms, or things like that, that when they never had these before, these have been known to happen in people with G6PD deficiency, either in reaction to drugs or even in reaction to virus. Oh, I should have brought up the study, and this may have been brought up on one of the other sessions that you did before. And sorry I don't have a slide on this, but in 2008 there was a study done on a very similar coronavirus called HCOV229E. Okay, and it is normally very mild, like just a mild cold in most people, okay? But there was an in vitro study done, and it showed that G6PD cells that were low in G6PD were extremely much more likely to get more replication of this particular coronavirus and the cells to die more than cells that had so-called normal G6PD deficiency. I mean, normal G6PD levels, okay? So this is why some doctors early on in the pandemic did start asking and saying, well, hey, we should look at this. We should look and see with COVID-19 if it's the same with SARS-CoV-2 as the same as HCOV229E or not. And let's just look at it and see one way or the other, prove or disprove, okay? Maybe two things could be done. One is to repeat the in vitro study that was done with HCOV229E. Do that for SARS-CoV-2, okay? That would be one thing. Another would be to do the correlation study, okay? To take people that are the most severely ill in the ICUs, etc., with COVID-19, and just see if we could find out their G6PD status, and if there were a whole lot more affected by COVID-19, that then what the general population would be expected to be. But of course, the problem with that is, as Keely raised, you can't really test people when you're already sick, okay? So then this is where the idea came, well, the military already knows everybody's G6PD status, and it's the most accurate testing, the cytochemical assay test, and they did it when they were healthy, so they're pretty sure this is accurate, okay? It should be in their records. So why don't we get like a military hospital or even a VA hospital, because a lot of them, they were in the military when it was first taken, when they got their status, so they would be, in the veteran system now. Or even at first, when I think it was early in the pandemic, was it in February or March, when that USS Roosevelt ship, there was a big outbreak on there, the Navy ship, and over 2,000 people tested positive for COVID-19, but only 8 had to go to the ICU and 1 died. Okay. Well, could there be kind of some information about what out of those 8 that had to go to the ICU? Like, was a disproportionate number of them with G6PD deficiency? Okay, so like people started asking these questions. And a doctor named Dr. Dan Vick wrote an article that appeared in MedPage. And he had some military background also. Then he was at University of Central Michigan, I think at the time when he wrote that paper, just kind of asking, hey, can we do the study? I think he appeared on a television show too, because in Syracuse, a local show. I guess he used to be from Syracuse originally. But still, nothing was done. And since then, he's written 2 more papers. One has been in the Mayo, written in the Mayo Clinic publication, and most recently in the British Journal of Medicine, that's on the World Health Organization website. And again, if you do a search for like G6PD COVID-19, there are dozens of articles and letters and case studies and what people saying this needs to be checked. Doctors and PhD biochemists saying we need studies on this. This needs to be checked. And yet nobody's been able to actually get a study done. And this is a major problem that, I would love to collaborate with people to figure out what we can do. I feel kind of discouraged because since I know Dr. Dan Vick has military ties and he hasn't been able to, it's like, what will it take? And especially since a lot of, the military is doing a lot of research, and I'll show you some of the slides in a minute of the other things. But Fort Detrick, I think, is the center right now of a military. I saw something pop up on there, but I couldn't read what it was. Was that something important for me to know?
No.
Okay.
That are commenting.
Okay.
I just put a comment sometimes.
Okay.
But
Fort Detrick, I think, is the center of doing the coronavirus, collaborating things. I'm sure there's other places also. And also the Pentagon funded to the tune of $40 million over the past 7 years. They funded the Eco Health Alliance and Peter Daszak, right? And studying pandemics, coronavirus, and the, what do they call it? Gain of function, how it jumps from one species to another and whatever. I'm sure, I would think that being that they've gotten so much money to study, this, that they must have been aware of this 2008 study on HCOV 229E. So like why that wasn't one of the starting points for COVID-19 is a baffling question. And I myself have tried, I had a professor that was very interested in getting a grad student to do some research on this. And so she contacted the head of research, I'm in Orlando. She contacted the head of research at the Orlando VA hospital. And she asked him, could she have a grad student analyze the data of like, how many people have you had in the ICU in the VA clinic, in the VA hospital for COVID-19 and compared to their G6PD status, okay? And she was really interested in doing that and said the grad student was interested, but she was told, oh no, they don't have time to help the grad student, because I guess they would have to help the grad student to see how to access the information or something like that. So these are the kinds of answers that we're getting. Now, most recently in the answer that Dr. Vick got in the most recent letter that he published in the British Medical Journal, it says that he approached people in the military back in the summer, and they said that maybe that they thought, oh, the sample size would be too small or something like that. Which, I mean, if you're talking about active duty who have died, it's small, and we'll go through the things to show that. But overall, you know, as I said, those who have been in the ICU, I could not find that information, but I'm sure they have it because they have everything else. So I'm sure it's enough to at least start a correlation investigation, and like why none of the others have anything. Okay. Do we want to go through the others? Are going to be just boom real quick. This is the only one there. So I don't know if you want to look at the others just to show, or if you feel like we don't have time. Do we have to wrap it up?
Well, we are coming up on 2 hours.
Okay.
I'll just say that well, not because I am a non-Hispanic Black, but mathematically those numbers jump out at me for the non-Hispanic Black group. Terry, do you want to quickly go through the last 3 slides, because you only have 3 more?
Yeah, just each one will be like a few seconds, really, not like minutes even. But Niloufar, did you want to say something?
Yeah, no, I just want to call out that I do understand, and Terry, like I said, is one of the more passionate members of our task force, that I think it's more awareness than anything else. It's people are not aware. They don't understand the implications of being G6PD deficient. And now the good thing is that there is much more research that's coming out every day. We see, we're seeing new articles and new publishers. I think we are not aware. There might be some countries somewhere that are already doing these kind of clinical studies, but we haven't seen anything except a recent research paper that did come up on the Sardinian population in Italy. That was a pretty interesting one. I think, Terry, you shared that. But we definitely need the clinical studies to be done to really understand what is that correlation, and also how have the drug treatments worked for this population. So it is important that we can get as much clinical studies published. Unfortunately, folks are not aware, and because we are mostly asymptomatic, this group is kind of I consider myself very healthy in general, but there are circumstances where these individuals need to be addressed a little differently, and that's what we're trying to champion. And I'm hoping that soon enough we get some sort of study done. The US military is the easiest place to go, given that they have such a huge database on G6PD individuals.
Okay. So do you want to just go to the other slides, and I'll just say a couple words about them?
Sure. Then we'll wrap up.
Right.
So
it doesn't matter what order. Okay. So this I just went to show that there are, if you Google COVID-19 and the military, there is a lot that comes up. Early on, they were saying that they were contracting it at a higher rate than the general U.S. population, but lately it's been lower rate that they're testing positive for it in the military. But it's a shame and I would have to look, because this is from May of last year, and I don't know if it's still the same, but it's saying here that they're banning coronavirus survivors from joining the military, I guess because of concern over some of the long term effects that we've been hearing about, the long hauler effects and everything. I don't know if that's still true or not. I would have to look into that. So, okay, the next one. Okay. Okay, so as you can see, the deaths this is in the active duty military only, not the VA, not the civilian or dependents, but they only had 4 die in November, and that was the greatest amount in 1 month, I guess. And then 3 in January. And it's hard trying to find out information about them. So it says, I just had something a day ago that said 2 just passed away, and the one they wanted to remain anonymous and they didn't give a name. The other one was a name I had never heard before, and a first name. And I looked it up, and it's like a Somali typical name in Somalia, from what I can tell. But again, that doesn't tell you one thing for sure or another. But again, it would be, even with the small number of deaths and ICUs, it would be good to know if we could find out the G6PD status and whether it was similar. Overall military, active duty military is only 2.2% overall. Did the ones who have died and the ones that had to go to the ICU, was it more than 2.2%? How much more? Questions like that. It wouldn't have to be like some people are saying, oh, it might violate privacy concerns. Well, if you're tying it with an individual person, yes. But to just look overall at percentages, it would not do anything, to violate privacy. Okay, the next one. Okay, and yeah, I just wanted to say, so the percent who have died in the military who compared to contracting it is a whole lot less than compared nationwide. Why is that? Well, it's probably a combination of things. One is because again, the active duty military is going to be the most healthiest cohort that there is, okay, to begin with. Number 2, if their G6PD status is known and they get, COVID and they get treated, they're not likely to be given a treatment that's contraindicated that could make them worse. That would be another thing. So those are just my thoughts there. And then who knows whether they have other things that they're also doing to help them, once they're in the hospital. I don't know. I'm not sure about that. And probably they have really good access to medical care too, as soon as they get sick, if they're on a military base or they're active duty, they don't really have to worry about, access to care or paying for it and things like that people here in the United States do. I mean, that aren't active duty military. But anyway, that's, my question for people to ponder. And if you want to look me up on Facebook or Twitter or anything else, that's fine also. Or if you have any thoughts about this, is like, is there any way we can get this information from the military? Like, what would be the best way to go about it? If people have any ideas, since again, they have the most accurate and complete records on this. Okay, that's it.
All right. Well, thank you, Terry. I think, yeah. On that note, we'll conclude. Lufer, do you want to say anything before I give my parting? I think my takeaway from all this is awareness. But go ahead.
No, I agree. So, Anwar, I was just sort of bringing up, reiterating Terry's point there, that since I'm aware that I'm G6PD deficient, one is having a very healthy lifestyle is important. And I've seen a lot of cases, while we don't have clinical data, we do have, I would say I have a handful of cases that I know of G6PD deficient individuals getting COVID and getting out of it fine. I do have a few cases where the outcome was not as positive, but we do need a comprehensive study to be able to decide what the outcome is. And like Terry was saying, maybe the treatment options are playing a huge role here, because if the US military folks are aware of their G6PD status and are being treated accordingly, that would indicate that the right treatment options for this community can actually make a huge difference. So that's a very interesting hypothesis. Again, we don't know what's happening, but it is a very interesting hypothesis. And overall, I think I'm with you, Anwar. It's awareness. It's getting more aware, and awareness for the general population, but also awareness for the scientific and medical community, and awareness at the policy level so that things can change. I think we can make a huge difference if we can get out there and get the word out. Sorry, I summarized before you, Anwar. Yeah.
Okay, well, Keely gave a thumbs up, so I think we're all good to go. All right, everyone. Well, thank you for watching. I know there will at least be one other discussion with the G6PD Foundation and Taskforce. Go ahead.
Sorry, no, just wanted to mention, unfortunately, Tivani was on. Tivani has a wealth of information. I think we should definitely plan a session with her. She was not able to, due to connectivity issues, we weren't able to have her on for too long. And then we do have other task force members as well who from all over the world that can, contribute. So yeah, I'm looking forward to a few more for sure, Anwar.
Okay. Okay. One thing, and I'll take us out on this. One thing I didn't mention in my intro was that my last 2 live streams actually on the vaccine, and the last one was on the COVID-19 vaccine. The last one I did focus on some of the adverse effects that were identified in the Pfizer and Moderna memorandum from the FDA. So if you're curious, that's been in the news quite a bit, and it'll probably be in the news going forward. So if you're interested in those are on the channel. But I want to thank Niloufar, Keely, and Terry for that wealth of information. As Niloufar said, we have to circle back and to try to do something with Tivani in the short term. So look out for that. And as I described when Keely got finished talking, I thought of some ideas that for some videos I might shoot just on some of the detection methods and screening methods that Keely talked about, some short pre-records. So with that, we will conclude. I don't know if I acknowledged him, but I want to thank Extended Clips for the Cash App donation. And if you have any questions, leave those. If you're watching on the playback, leave those in the description box below. Please like, please share, and also please subscribe. Everyone be safe out there. And as always, remember that your attitude determines your altitude. Take care, and I'll talk to you the next time. Bye-bye. Let's see. Yeah.
Thank you, Anwar.
Okay, Niloufar. Keely.
Thank you.